Okay class, in this video I'm going to go through part 2 of unit 1. So we're going to start by going over a brief overview. You should have already learned this in biology. We're going to do a brief overview of cells and the different organelles within a cell. And then we're going to talk about some chemistry, basic chemistry concepts. Okay, and I'll try to let you know like why you need to know these things for A and P as I go along. Okay, and why I'm doing a review of these things. Okay. Okay, so animal cells have a plasma membrane that surrounds the cell, and it's a lipid bilayer, right? That is semi-permeable, so it allows certain things in and certain things out of the cell, right? We don't want to let anything into the cell, right? You hopefully don't want to let like bacterial cells or virus cells or viruses into the cell, okay? And you don't want to let like all of your water out of the cell, for example, okay? So semi-permeable membrane, okay? And we'll talk about that a lot throughout AMP 1 and 2, okay? Like when we talk about how a neuron fires and certain things are let into the cell via different channels, okay? Or action potential of a muscle cell as well. Okay. All right, so within the cell, we have a cytoplasm. So that's just the whole interior of the cell. So that includes like all of the organelles, the cytoskeleton. Okay. Cytosol, this is the fluid portion of the cytoplasm. So there's like a gel-like fluid within that helps keep all of the organelles where they need to be. Okay. So again, the plasma membrane is selectively permeable and only lets certain things in and certain things out, okay? So a concentration gradient, this is the difference of concentration of usually an ion, but it could be anything, right? A chemical as well, inside versus outside of the cell. So if we have like our little basic cell here, if we have a lot of sodium outside of the cell, a lot of sodium here, but we only have a little bit of sodium inside the cell. Its natural concentration gradient, it's more concentrated outside versus inside, it's going to want to move into the cell, okay? So through its concentration gradient, it can usually move naturally into the cell without a lot of help, okay? If it wants to move against its concentration gradient, so if it wants to move out of the cell for whatever reason, again, when we get into these things, we'll talk about that, then it would need help, right? So it needs some sort of active transport, which is where it gets help by another ion or through the help of ATP or energy, okay? An electrical gradient is the difference in concentration of specifically ions. So these ions, this could be a concentration gradient or an electrical gradient. These ions create a sort of a charge, right? So that you can measure with like a voltmeter. Okay, so things want to move toward their electrical gradient as well, okay? And then together they can make up an electrochemical gradient, right? So they can kind of work together. And we'll talk about that more, again, when we get to like the nervous system, okay? So I talked about this a little bit, but transport can happen through passive processes. So simple diffusion, so if it's moving like with its concentration gradient, like if the sodium wants to move in, that can happen via simple diffusion. So it just diffuses through, okay? And usually small ions and things can move through the lipid bilayer, okay, or the plasma membrane. Or osmosis is specifically the movement of water, okay? Or through active processes, and again, these require help of another ion or they require energy, usually in the form of ATP, okay? All right, so osmosis is movement of water. So if we had some sort of system where we have a solute, like sodium on one side and just water on the other, so say we have like this U-shaped tube here, okay, and say we have a lot of, I'm going to draw my sodium ions in red here, this is our selectively permeable membrane here, so only so sodium can't pass through it, but water can. Okay, so we have a lot of sodium and water on this side, and then on the other side we'll draw our water in blue. So we have water in here as well. But on this side we have a lot more water because we don't have that sodium solute taking up the space. The water is going to naturally move via osmosis to the area of the higher solute concentration. Okay, so it's going to move this way, and then you're going to have a lot more water within here along with your sodium and the little bit of water you already have. Okay, so that's osmosis. It's just movement of water to an area of higher solute concentration. Okay, tonicity. This is important when we talk about red blood cells. Again, this will be an A&P too, but I do want to give you an overview of it. So it basically relates to how water influences the shape of a cell. So red blood cells have are a biconcave disc shape, so they kind of cave in the middle on both sides and then kind of pop out around the ends. Okay, so in an isotonic solution, okay, there's an equal amount of salt and water concentration inside versus outside of the red blood cell, so it keeps its nice shape. Okay, if we have a hypotonic solution, that means there's a lower concentration of like sugars or salts compared to blood. Okay, in that case, water is going to enter the cell and the red blood cell can burst. Okay, and then we have hypertonic solutions. Okay, so this is a higher concentration of sugars or potentially sugars and salts than there is blood. So in this case, water would leave the cell and the red blood cell would become dehydrated. Okay, so it loses its shape, it kind of gets smaller and dehydrated. Okay, so neither of those are good, hypo or hypertonic. We want more of an isotonic solution, or we want our blood to be more isotonic. So that our red blood cells maintain their shape, and their shape is helpful in how they deliver oxygen. Okay. All right, so back to the different organelles. So the nucleus, this is what holds our DNA. Okay, and this is what gives the instructions for everything within our body. Okay, so it has surrounding it what's known as a nuclear envelope that has these little pores. So DNA cannot leave the nucleus, but things can enter, or RNA can leave the nucleus. So RNA is copied from DNA, and then RNA could leave the nucleus and create proteins within our body. Okay, and then proteins do lots of different things. We'll talk about various different proteins throughout A&P. Okay. Okay. So it's like the control center of the cell. Okay, the endoplasmic reticulum, we have, it's like a system of these internal membranes. Okay, so we have rough endoplasmic reticulum, which have these little like granules in them, which are ribosomes. Okay, and ribosomes are where proteins are created. Okay, so RNA comes into the ribosome and a protein is created within the ribosome. Okay, so those are very important. And then there's a smooth ER, which does not have ribosomes embedded within it. Okay, so they like make things within the cell, like proteins, right, in the rough ER. Okay, smooth ER, that's where carbohydrates and lipids are created. Okay, so proteins are made in the rough ER, smooth ER, carbohydrates, and lipids. After things are made within the endoplasmic reticulum, they are packaged and sent out via the Golgi apparatus or Golgi complex. Okay, so maybe you learned this like in high school or in your previous biology class, but it's like the UPS of the cell. It kind of packages things into vesicles and ships them to where they need to go, either somewhere else within the cell or outside of the cell. Okay, so Golgi complex. So they work together. Okay, there are also things like lysosomes found within different vesicles, and lysosomes lyse things, which means like to cut them up. So lysosomes will break down any sort of like trash within the cell, or if there's like a bacteria or a virus within the cell, it can help to break that down as well. Okay. So cell cycles, we're going to talk about the basics of, again, this should be a review, mitosis and meiosis. Okay, so mitosis happens within our somatic cells. So all of our cells except our gamete, egg, and sperm cells. Okay, meiosis is how those replicate. Okay, so first let's talk about mitosis. Okay, so before we get to mitosis, we have an interphase, okay, which has 3 different phases. Okay, so interphase has a G1, S, and G2. Okay, these G phases are growth phases. S is like a synthesis phase. Okay, so G1, this is where the cell actually spends most of the time of its life, and it's just like growing into its normal size. S phase, this is where DNA synthesis occurs. So before a cell can copy itself, in order to have the same number of chromosomes in the mother cell and the daughter cell, it needs to double its DNA first. So synthesis is where that DNA is doubling. And then G2, this is just the cell further preparing for cell division. Okay, so this is where mitochondria is replicating and microtubules are starting to be synthesized. Okay, so this is all within interphase. Okay, then we have mitosis. Okay, mitosis, we have PMAP. Prophase, metaphase, anaphase, and telophase. And then finally we have cytokinesis. Okay. So first we have prophase. This is where the nuclear envelope starts to disintegrate because we need access to our DNA. Okay. The chromosomes start to become visible. Okay. And the centrioles are starting to move to opposite sides of the cell, so we're kind of preparing for that cell to divide. Okay. Metaphase, this is where the chromosomes line up. So here, I'll draw a little cell. This will be the nucleus. So the nuclear envelope is starting to disintegrate there. Okay. And then our DNA kind of becomes visible within there. Our centrioles start to move to opposite sides of the cell. Okay. Then we have metaphase. So here we have our chromosomes lining up in the center of the cell. Okay, metaphase. Anaphase, I'm going to draw over here. In anaphase, our sister chromatids, so each chromosome has 2 copies, our sister, known as sister chromatids, they separate and move to opposite sides of the cell. Okay, so how metaphase happens is the centrioles create microtubules that push the chromosomes to the center of the cell, and then they start to disintegrate and they pull the sister chromatids to opposite sides of the cell. Okay, and then in telophase, now we have what's known as a cleavage furrow right here, where the cell is starting to pinch into 2. Okay, this is sometimes referred to as like the opposite of prophase. So the nuclear envelope starts to reform around the DNA, okay, on each side, and then this cleavage furrow forms, so the cells are starting to pinch apart into 2 cells. And then cytokinesis, now we have 2 identical daughter cells, okay, with the same DNA as each other. Okay, because remember, in interphase, we doubled our DNA and now we're dividing it again, and it's the same genetic material that was copied. Okay, now meiosis, this is how we go from, or how we get more egg cells or sperm cells, so those gamete cells. Okay, Here we made what's known as diploid cells, okay, because they have both copies, like di meaning two. They have both copies of each chromosome. Haploid cells are made from meiosis, okay, and haploid cells have only half of the genetic information of a somatic cell, right? Because sperm and an egg cell come together to make a zygote, which would then have all of its genetic information, okay? So you only need half of the genetic information in your sperm or egg cell, okay? So there's two different divisions in meiosis, so let's go through those. Okay, there's meiosis 1 and meiosis 2. So a few different things happen from mitosis to meiosis. So first, before meiosis 1 begins, there is a doubling of DNA, okay? So let's say our starting egg cell has, it's more, but let's just make it simple and say 2 chromosomes. Before meiosis 1, it doubles to 4. After meiosis 1, we cut it in half to 2. Okay, so this is like our normal somatic cell number, let's say. It's not really 2, it's a lot more. Then after meiosis 2 occurs, it's going to be another separation. We get to 1. So we get to half of the genetic information from our original cell. Okay, so this would be our 1, our sperm or oocyte or egg cell. Okay, all right, so meiosis 1. Okay, so we have the same thing, PMAT1, and then in meiosis 2, it's PMAT2. So prophase 1, metaphase 1, anaphase 1, telophase 1. Okay, and then in meiosis 2, we have PMAT2. So prophase 2, metaphase 2, anaphase 2, telophase 2. Okay, so a couple different things happen in meiosis, okay. So in prophase 1, we have our DNA. Let's say this is just the nucleus here, okay. So we have our chromosomes here. Let's draw another different color chromosomes. Okay, so they line up near each other, the different chromosomes, and they actually switch some of their genetic information with each other, okay? So this is known as crossing over, okay? So then we're left with something like this. And this, for example, okay? So now this is one of the reasons why we don't look exactly like our siblings, even if we have the exact same 2 parents, okay? Because we want genetic variation, okay? We don't want to keep passing down things that are like negative, for example. So we want lots of genetic variation, okay? So this happens during prophase 1, okay? So they pair up and exchange segments. You still also get the disintegration of the nuclear envelope, the chromosomes becoming visible, all of that, okay? Metaphase 1, the homologous chromosomes align along the central plane, okay? So metaphase 1 are different chromosomes with now our exchange genetic information, and they line up randomly each time. So that's another way we get more genetic variation, because they line up in a different random way each time. Okay, metaphase 1, we have our full chromosomes. Anaphase 1, our whole chromosomes move to opposite sides of the cell. So we have, let's say, 3 of our chromosomes move to this side, 3 of our chromosomes in our example move to this side. Again, it's a lot more than 3. Okay, so they move to opposite sides of the cell. And then in telophase 1, now we have 2 daughter cells, and they're going to have different genetic information. Okay, so different genetic information from each other. So these are not going to be like sperm cells that have the same genetic information, will have a little bit different genetic information. Meiosis 2, this is basically like a mitotic division. Okay, so in prophase 2, now we're starting with 2 cells. Okay, nuclear envelope disintegrates and our chromosomes become visible. Okay, metaphase 2, our chromosomes line up in the center of the cell here. Okay, and then anaphase 2, now our sister chromatids separate to opposite sides of the cell in both of our cells. Now we have 2 cells, right, from meiosis 1. And then telophase 2, these are all going to pinch apart, and then we're going to be left with eventually 4 cells, all with different genetic variation. So after this, we get our 4 cells, each with different genetic variation. Okay, so these would be 4 sperm cells, for example. Okay, and this will become important when you learn about the reproductive system at the very end of AMP2 and how spermatogenesis and oogenesis occurs. Okay, so we're not going to get into that now, obviously. Okay, and then we're just going to get into some basic principles of chemistry, and then we'll be done with Unit 1. Okay, so chemistry is how matter interacts. Okay. So matter is anything that has mass and takes up space. So this includes atoms as well, which we learned about a little bit in the previous video. Okay. So all elements have chemical symbols. Okay, like from the periodic table, right? Like oxygen is O, carbon is C, hydrogen is H, iron is Fe. Right? These are all important things within our body. Okay. Ions, these are things that have a charge, so either positive or negative charge. Like sodium has a positive charge, potassium has a positive charge. Chloride ions have a negative charge to them. Calcium has 2 positive charges. Okay, so ions, anything that has a charge to it will get into a lot of different ions. A chemical reaction, this is when new bonds are formed and or old bonds are broken. Okay, so for example, to make water molecules, we have our 2 hydrogen ions, which are bonded like this, okay, because they're H2, so these are like 2 hydrogen molecules here, okay, so we have 2 H2 plus our oxygen, okay, and these bonds break here and they form H2O. So we have our oxygen bound with 2 hydrogens. Okay, so now we have our 2, here's our oxygen, and then we get 2 water molecules. Okay, so it's a very simple chemical reaction, adding hydrogen with oxygen and getting water, H2O. Okay, and we'll talk about different chemical reactions. Okay, acids, bases, and salts. So acids are things like hydrochloric acid, for example, has this hydrogen. This hydrogen will dissociate or break apart in water. Okay, so it dissociates in water. The more it breaks apart, the stronger the acid it is. Bases have, it's like potassium hydroxide, have OH groups, okay, or hydroxide groups. with bases, hydroxide groups dissociate in water. Okay, the more it dissociates, or the stronger the dissociation is, the stronger the base you're talking about. And then salts have two ions that break apart, like potassium chloride. In water, these will both break off, and you'll have these two ions within your water. Okay, so that's a salt. Okay, and it'll be two ions that are neither that don't include hydrogen or hydroxide. Okay, so to tell how strong an acid or how strong of a base we have, there's a pH scale. The pH scale goes from 0 to 14. Okay, the lower on the pH scale, the more acidic something is, and the higher on the pH scale, the more basic something is, also known as alkaline. Okay. A neutral pH is right in the middle, around 7. So this would be neutral. Okay. So our blood is about 7.4. pH of about 7.4. But our stomach acid is about a pH of 2. So very, very acidic. Things like household cleaners tend to be more basic with a very high pH. Okay, and in the PowerPoint there's some examples so you can go through those. Okay, some common things that you find in the body. We have carbon. So when you talk about like organic chemistry, it has to do with molecules with carbon atoms. So organic compounds, they always contain carbon. So carbons can combine in lots of different shapes and make like ring structures. So those would be like sugars, or they could be like long chains, like carbohydrates. So they're a good source of energy, right? Carbohydrates are where we get all of our energy from. Okay. Proteins, these give structure to the body. They help to regulate different processes in the body. They make up enzymes, which we'll talk about. They help in contraction of muscle. And they help to transport different substances. Okay, amino acids are what make up protein, so they're like the building blocks of proteins. You probably heard, right? Proteins are formed by combining different amino acids together in different ways. Okay, and then the protein folds up. Okay, so it has like these alpha helices, which are kind of like ribbons that go like this, beta pleated sheets, which are like folded pieces of paper, and then those all kind of come together and fold up in a certain way, and once it's folded up, then the protein can be used. Okay, enzymes, these are catalysts. These are used to make reactions go through in the body. Okay, so they're highly specific, very efficient, and subject to different cellular controls. So let's say we have our enzyme. Okay, so we have our enzyme here. Again, an enzyme is just a protein. This here would be the active site of the enzyme. So this is where something can kind of come in and the reaction can occur, and the enzyme helps that reaction to occur. Okay, so say we have sucrose, which is a sugar molecule. It goes into this active site, and this active site is very specific, so only certain things can fit into this active site. So in this case, sucrose. Okay, then the reaction occurs. What really happens is this protein undergoes a conformational or shape change, and we break up that sucrose into fructose and glucose, okay, so into smaller sugar molecules. So it leaves this enzyme, and now our active site is open for another sucrose molecule to come in, right? And we need our glucose by itself because that's what we break down into energy in the body, or what we use to create energy in the body. Okay, nucleic acids, these make up DNA. Okay, and DNA forms the genetic code of our body cells. Okay, so it regulates most of our cells' activities, and RNA is made from DNA. Okay. So some differences of RNA versus DNA. DNA is double-stranded, it has 2 strands, whereas RNA is just single-stranded. Okay. DNA encodes the information for making proteins, whereas RNA just carries that genetic code and then assists in actually making the protein. So again, the RNA goes into the ribosome and creates a protein based on the code it was given from DNA. DNA stands for deoxyribonucleic acid. Okay, so it's missing oxygen on one of the carbons on the sugar. And RNA stands for ribonucleic acid, so it has an extra oxygen on one of the sugar carbons. Okay. The nitrogenous bases are the same with DNA and RNA. 3 of them are the same, one's different. Okay, so adenine, cytosine, and guanine are the same, but DNA has thymine and RNA has uracil. Okay, and they're like interchangeable with each other. Wherever you have thymine on a DNA, you would have uracil on an RNA. Okay, so in DNA, adenine base pairs with thymine, and in RNA, adenine base pairs with uracil instead. Okay. So you should know the very basic differences between DNA and RNA. And then finally, ATP. Okay, ATP has an adenine bound to a ribose sugar and then 3 phosphate groups. So this is where we get the majority of our energy in the body from those phosphate groups bonds breaking from each other. Okay, so going from ATP adenosine triphosphate to ADP adenosine diphosphate. Okay, so we'll talk about when that happens in different units throughout AMP1 and AMP2. Okay, so that's it for Unit 1. I know it was a lot of review. In the next unit, we will get more into detail of different tissues and then integumentary systems.